All right. Thank you very much for joining my session on prefilled syringes. I have the real pleasure today to give you some insight how we at Fresenius Kabi support our customers to manufacture prefilled syringes, not just on the small format syringes, such as the 1 milliliter, 2 or 3 milliliter, but up to 50 milliliter. And then on top of that also, we are very active in the field of complex formulations. So that means that we have some capabilities to formulate emulsions, liposomes, oil-based formulations. So I would like to give you some glimpse what it takes to do those complex formulations. Before I dive into that, just a few words about myself. So my name is Franz Kainz. I'm heading the global contract manufacturing business within Fresenius Kabi for sterile pharmaceuticals. I'm a molecular biologist by training, and over the last 9 years or so I've focused on contract development and contract manufacturing of steroids, and also to some degree I focused on drug delivery technologies. So in my talk, I would like to give you a first idea, like what we as Fresenius Kabi do. Before then, I make somehow a deep dive, starting from the manufacturing plant to commercial manufacturing to, at the end, to development services for prefilled syringes. So Fresenius Kabi, for those of you who do not know our company, it's a very, very large company. So more than 270,000 people are working for Fresenius. For Fresenius Kabi, more than 33,000 people are working for that company. And we at Fresenius Kabi focus specifically on sterile pharmaceuticals mainly. So to give you an idea about the different technologies, we are active, of course, in the field of IV drugs, which is, of course, also the choice usually for prefilled syringes. At the same time, we also do some other technologies such as clinical nutrition, also for IV administration. And the reason why I mentioned that is that this also is somehow linked to the complex formulations, because we have emulsion technology within our company. And an emulsion for IV administration is pretty hard to do. And these emulsions, for instance, can also be used for drug delivery of oil-based formulation. So if you ever had an operation and got anesthetized, the chances were high that you were anesthetized with propofol, which is one of the main products also for Fresenius Kabi, which is actually a lipophilic API dissolved in an emulsion. Yeah? And based from that history, from that pedigree of emulsion, we used that technology also to go into liposomes. We also have implemented suspension technologies, and all those technologies are very interesting then also to combine it with prefilled syringes. So a good example is that we actually also fill propofol in 50 milliliter syringes, and that's a technology, of course, that we also offer to our customers to formulate those complex formulations and fill those formulations in those large syringes. Infusion therapy is, of course, a big topic. So my colleague Benedikt Reichert will also give a talk about other containers like bag technology. And so if you're interested in that, please also join the session after me. The point that I would like to take here with the infusion therapy is that when you use, for instance, a 50 milliliter syringe, then this is also a very, very interesting technology then for controlled administration, because you can attach or you can put the 50 milliliter syringe into a pump, and therefore you have a very precise control over that. And last but not least, we are also active in the field of biosimilars, so we also offer the fill and finish services of syringes, of biologicals in syringes, and therefore it requires a lot of other expertise, such as yield, for instance, You really want to make sure that every little drop goes at the end in your syringe and is not wasted during the process. So within that portfolio now, we take advantage of our manufacturing sites. We operate more than 20 manufacturing sites worldwide, and take advantage of those technologies and add value to our customers by offering contract manufacturing services. So I'm heading that business within Fresenius Kabi, and I'm really proud of that we have a lot of different containers within our portfolio. So my talk today is about syringes. I don't want to spend too much time about the other containers such as vials, bags, and ampoules and bottles. Just in case if you're interested in bags, please join also the session of my colleague. We operate a global network of manufacturing sites, mainly in Europe and US, but also in Asia and in South America, and one in South Africa. I've highlighted already the 2 sites in orange where we do pre-filled syringe, because the container pictures that I've just shown you before are a good representation, what we can do on a global basis. At the same time, when it comes to syringes, we only have 2 sites where we offer that technology. And that's, on the one hand, the Graz site in Austria, and on the other side, Wilson, close to Raleigh in North Carolina in the States. So let me now take you to a journey starting from the manufacturing side, and then we go to the filling line, to the specific compounding process, and to the development. So basically a zoom in starting from the plant. So hopefully the battery is not going. Okay. So the site in Wilson is an interesting one in such a way that it's initially a site that was only focused on syringes. So we currently have around 260 people working for that site, and have multiple filling lines there for pre-filled syringes, with the main focus on that plant for the small syringe lines, such as the 1 milliliter, 2 milliliter, and 3 milliliter. We are even thinking about it also to expand it to the larger formats that will come in a second. At the moment, however, we are focused on those small formats. We can do controlled drug substances there. We have a setup of disposable equipment, also multi-purpose equipment. And when it comes to the compounding, we either use actually fixed tanks if the product allows it, or we actually take advantage of mobile tanks. The main focus on in Wilson is actually aseptic manufacturing, even though that we have terminal sterilized products also implemented. In terms of visual inspection, it's interesting that at our Wilson site we have the capability to do 100% automatic inspection, whereas in Graz, which I'll show then in a few minutes, we only actually do a semi-automatic visual inspection. What that means, I will then come later during my talk. The site is currently approved by the US FDA, even though that we would also have the option to expand it to the European market and so on as well, because the plant fulfills all the requirements also for the European market. The Austrian side is a bit different, that we do not only do syringes there, but we have the whole range of containers, such as vials, ampoules, and bags as well. The site is pretty large actually, so more than 900 people are working there. On the top you see the site, which is only the site for the filling services, whereas the packaging facility is almost the same size as the top picture here, where we do actually then the packaging. So we fill currently more than 100 million units of all different types of containers. And the interesting thing here is that we have those interesting formulation technologies. So that's also the title of my talk, complex formulations. And we fill that in those different formats up to 50 milliliter syringes, which makes us in a rather unique, which puts us in a very unique spot from my perspective. And in terms of technologies now here, we would do aqueous formulation, oil-based formulation, emulsions, suspensions, and lyophilizates. Having said that, we do not currently do lyophilizates in syringes, but all the other formulation technologies at this point. The plant is actually one of our competence centers for aseptic manufacturing. And this is, of course, an important point because, I mean, usually when you choose a syringe, you would only do that for a value product, right? And we see now that trend towards biologicals, towards really expensive APIs, and therefore those APIs are usually also often manufactured aseptically, so they are not suitable for terminal sterilization. The nice thing is also that when you actually develop a product, you want to start at a small scale. And therefore, we also invested, I believe 15 years ago, in a pilot plant where we also do the process development, but can also fill the product in syringes on a small scale before we do the scale up on a commercial scale. The site is approved by US FDA, EU and Asia, so it's a true global manufacturing site for global supply. So what I already announced before, let's do now a deep dive into the filling. So we operate an optimal line at Graz, which is a full isolator line. We use only nested syringes. So nested syringes, for those of you who are not familiar with it, simply means that we order the syringes already in an aseptic way. So they come in a tray, are pre-sterilized and ready to go for filling. In terms of filling technologies, it very much depends which formulation you have, right? If you actually have an oil-based formulation, we would usually rather use rotary piston pumps, whereas if you have an aqueous formulation where speed is really important, you would go for peristaltic pumps. So you have advantages and disadvantages using both systems. Very much, of course, depends on the formulation. The same is, of course, also true for the stoppering technique. So insertion tubing is a way that you actually have a certain tube where you attach then the stopper to the syringe, or for the small syringes, you would use a vacuum principle, which has the advantages that the air bubble in the syringe is then much, much smaller. And those things matter really a lot in the case, for instance, you want to use that syringe for an auto injector, because then you need to have a very controlled air bubble in order to have the same pressure with the auto injector. And for complex formulations, you often have the situation that the API is then oxygen sensitive, so we also have the option here to fill our syringes under nitrogen flushing, so to really make sure that there's no degradation of the API. Okay? And depending on the product, you would either go for dedicated equipment or disposable equipment. Very much, of course, depends on the API, and it's a case-by-case decision, right? The line is operated within a classroom C environment, and of course the isolator is then classroom A. So the filling takes place within a classroom A isolator. So going now deeper, let me just then give you a bit of an impression how that process then works. So we get those nested syringes in those trays that I've mentioned here on the left, right? And in Graz, this is a rather manual process. So the outer protection bag is then removed manually, and then within the isolator already, the Tyvek, so the protective foil, is removed. Then the tray is put on the conveyor belt, and all the syringes are then filled at the same time, filled simultaneously. So that very much also depends. If you have a 50 milliliter syringe, then you actually have less syringes in a tray than you actually have with a 1 milliliter syringe. And even though that the lines in Wilson in the US and the line in Graz are somehow similar, we have some differences in terms of automation. So the US lines are much higher automated, meaning also that removal of the Tyvek would be fully automated. It has some disadvantages as well, right? Because if you have, for instance, smaller batch sizes and different formats, then you also then lose some efficiency, of course, with the different products. Then the next step is, I think I also have a small animation here. So the next step then after the Tyvek removal is then the filling, right? Filling is then done under nitrogen flashing, what I already described if the product requires it. Then stoppering takes place with the different techniques, right? Either with the tubing or with vacuum. And if the product can be autoclaved, which is then like, a requirement, for instance, I will come to that also in a minute, then we would then put that in those large trays, right? These are some examples of those 50 milliliter syringes. And then afterwards it would be transferred to the other facility for packaging, where in Graz we would do the visual inspection semi-automatically. So what does that mean? It really means that all the handling of the syringe is done then by the machine, whereas the visual inspection by the operator is still manually. So basically you have an operator sitting there doing the inspection, and this is actually quite a tedious job. So you need to be extremely focused to spot everything. And from my perspective, when it comes to aqueous formulations, you can actually go to that automation step that you have a full automation. When it comes to complex formulations, such as emulsions or let's say also oil-based formulation, then it might be a bit more difficult to have that fully automated without an operator. Then the final assembly takes place fully automated with the rod attached to it, labeling, including of course also then if required, then the sterilization and blistering packaging depending on the product. So I have a slide on that as well, where I would like then to describe you also what the different options are in terms of packaging. And then very much depends, of course, with the finished product where the sterilization level is, right? If it's in a whole box or. But I think this was also a major project within our plant over the last years to get actually sterilization right. And here we would have then the option to offer either an in-house solution or partner up with TraceLink, what many of our customers actually did. Okay. So let me now just pick out a few examples of some considerations for commercial syringe manufacturing. So since the syringes come in those trays, right, pre-sterilized, we can have a lot of flexibility here, meaning that we can fill syringes with a Luer cone, with a Luer lock adapter or stack needle. So as long as those syringes are supplied in those trays, we can fill it on that line. And of course, there are a lot of design decisions at the beginning of a development, right? Either you want to go for a glass or for a plastic syringe, the different shapes of the syringes, right? In terms of formats, I think that's already the important point that I mentioned in the beginning, that we can go up to 50 milliliter, which is really a very nice thing to have. And we pretty much operate and cooperate with the major syringe suppliers, right? I think there's no company out there that we are not dealing with, because it very much, of course, also depends on our customers, on the preference, which syringe supplier they want to go for. So when you compare now glass and polymer, and of course, I mean, this is a bit subjective as well, right? Try somehow to compare the two options, right? Do you want to go for a glass or a polymer syringe? And I have to say, there are actually quite a lot of advantages over the plastic syringes compared to the glass ones, but it very much depends, of course, what are the product requirements. So like absence of heavy metals is one example, right? Because you always have the leachables from the glass, right? Then breakage resistance, I mean, that's an obvious one. When it comes to the design space, you can control the shape of the plastic syringes much better. And remember, as long as they're supplied in those trays, we can, of course, manufacture that. Oxygen protection is, of course, a tricky one, right? Because there are a lot of APIs that are oxygen sensitive, and the plastic syringe would not be a really sufficient barrier for oxygen protection. And in order to overcome that, we actually utilize blister technology, where the blister is then also in a nitrogen atmosphere in order to protect the drug product from oxidation. Then the glass syringes actually have integrated Luer lock. The glass needles have a Luer lock attached to the glass, so it's glued in, which is an issue then in terms of leachables, whereas the plastic syringes are made out of one piece. So I would also say that the plastic syringe wins in that comparison. And I mean, I think the disadvantage maybe a few years ago was that you didn't have that many suppliers out there that could supply plastic syringes. That has changed also over the last years. So the plastic syringes are on the market since probably 15 years, I would say, and now you really see more and more companies offering plastic syringes. And in terms of tight tolerance, so if you need to have a very precision in terms of tight tolerance, then the plastic syringes is almost a factor of 10 better than the glass syringes as well. At the end, I mean, it's a design decision, right? And I mean, it's very difficult, of course, to say like which syringe is better. We work with both, and yeah. And I just thought it's somehow useful, like to have some sort of comparison between the two materials. A few words about autoclaving and visual inspection, just very briefly. I mean, the guideline clearly says that if the product is terminally sterilized, you need to go for that, right? And that's also the reason that we have implemented both procedures at our two manufacturing sites, even though that the Wilson site is focusing a bit more on aseptic. But it's very, in the Graz side, I would say it's 50-50 between terminal sterilization and aseptic manufacturing. And visual inspection, I want to keep it short here, I already mentioned it. So the Wilson side is 100% automated, whereas the Graz is semi-automatic. This is now an interesting one in terms of the packaging option. So this is just an example of the 50 milliliter propofol syringe, which is oxygen sensitive. So therefore we have that blister for the oxygen production here on the A slide. We have implemented all the three options at our sites, like single carton as well. The flow bag is very, very popular. So the flow bag has the advantage that it's also a protection for oxygen protection. Yeah? And the other thing is also that space in the hospital is, of course, very limited. So if you actually want to have a very small footprint, then the flow bag is a very interesting way to pack the syringes. In terms of secondary packaging, so I have to admit, we of course do that on a very large commercial scale. At the moment, we do not yet have an auto injector technology implemented. So for those customers, we would do then bulk manufacturing, right, and then ship the bulk syringes then to a sub-supplier. But apart from that, we would offer the full range, right? Assembly of plunger rod, assembly of safety devices according to our customers' needs, and of course also implement the right sterilization solution to our customer. So I mentioned in the beginning that we are very keen to go into that field of complex formulation, and for complex formulation, I would also consider, like, utilizing high potent APIs. And if you think about that, like, high potent API, you don't want to take the risk in a hospital setup, for instance, to have a lot of handling with high potent APIs. From this perspective, it makes sense to do the filling right away into a syringe, because then you add at the end value. I mean, a syringe at the end of the day will become more expensive than if you do the filling in an ampule or in a vial. Having said that, there are also some risks, right, handling high potent APIs. And so let me just actually describe you some of the things, how we address the topics, how we process high potent APIs within our facility. We use a risk-based approach, meaning that when we compound a high potent API, the biggest risk is really when you have dust formation, right? And as soon as it's in a liquid formation, then the risk becomes much, much smaller. And therefore, we invested in a very nice Isolator Technology where we can go down from an occupational exposure limit to 0.1 microgram per cubic meter. So to give you a bit of an idea what that means, so if you think about a coffee sweetener, right, that's like a coffee sweetener that you put into your coffee, weighs probably 1 gram, and if you dissolve that in 10 Empire State Buildings, then you would be exactly at that limit where we are with our Isolator Technology. It's pretty impressive, I would say. Yeah, and once you're then at the filling level, then the risk becomes much, much less, because then you don't have the dust formation anymore, right? And then, of course, we have certain procedures in order to actually handle also those rather, dangerous APIs to protect our employees. So just a few more words about that Isolator Technology. I mean, I think we can handle actually also oxygen sensitive product. So the isolator is designed in such a way that it's run under both positive or negative pressure, depending of course on the API. We would basically not use negative pressure if it's not a high potent API, but then you really want to protect our operators from it, right? Then for oxygen sensitive products, we would flood the whole isolator with nitrogen, right, for protecting the API. And I think it's also really important that how do we actually bring it then from the compounding again to the filling line. And here we would either use mobile vessels, what I've shown here on the bottom, or we would also then use, for instance, a bag system. And that even goes then further, because you also need to make sure then that you also then dispose also any waste in the right container, so that there's no risk for our employees. Yeah. I think I already made the main points here, so I would just then move on. So basically, you take that mobile vessel, and then you attach it then to the filling line, or alternatively, for very expensive APIs, for small batch sizes, we also have the option that you simply attach one of those bags, right, disposable bag systems, to the filling line. And the advantage of such a system is that you minimize really the distance between the compounding bag to the filling line, right? Every drop matters when you fill a biological. Okay, so for the sake of time, I would then just move on. Just take some time. So let's have some consideration also when you move away now from the commercial manufacturing. Let's go down even one step deeper to the step before. What happens when you do the development? What things do you need to consider? And by all means, this is not, of course, a complete picture. It's just a bit of a glimpse of some considerations that are important for developing prefilled syringes. And one of the most important things is that as the applicant for a new drug product in a syringe, you also need to take into account that you're responsible for the syringe, which is by definition a medical device. And that's a very critical point. So not only the CGMB guidelines are important, but also the guidelines for handling the syringe. In other words, you need to make sure that when the nurse in the hospital or the doctor, uses the syringe, that all the consideration of applying the force and so on are taken into account for the development, and it's also then at the end of it part of the entire registration package. And for that, and at the moment it's like this, from a regular perspective, you have already then that regulation in the States, it's also going to be expected that the same combination product regulation is going to come to Europe. So from my perspective, I mean, combination product requirement is absolutely a must these days. And we invested here also in specific lab equipment which we use not just during the development phase but also during the release process. So this is like a machine from Zwick Roll, for instance, which allows us to test the different forces, like the pull pressure measurements, the torque measurements. For instance, let me just also have a quick look at the list. So for instance, you need to make sure that you know what's then the force that would result in the breakage of the flanger, right? Also then, when you take a syringe with the lure cone and when you attach then a medical device like a transfer set to the syringe, when would the lure cone break? So this is basically a setup, a machine which allows you to do that, and this is then also then the standard that we would actually take into account during the development. When it comes to formulation technologies, I mentioned that list of aqueous solutions, oil-based formulations, emulsions, liposomes and suspensions already before. I think one could give a talk about every single of that formulation. I think for me, the point is somehow that we used our long history on the emulsion technologies to move into those different areas. And if you think about now, RNA technology, we see now a huge burst of, using liposome technology for the new biological entities. And that's, from my perspective, a very exciting thing in the industry, where I also believe that we can add some value. So we would usually take somehow a toolbox of different development tools, right, depending, of course, on the product profile. And then do a design of experiment study, if required, for instance, and test all critical process parameters in order to make sure that during commercial manufacturing you really have then also control over the process parameters, rather than running 3 process validation batches and hoping that all the variation during the commercial manufacturing does not have an influence on, right? So, and this is exactly what I just described here. So this is just the toolbox on the left. Like the font is a bit small, but I mean the point is just we have a lot of different assays that we use during the development phase in order then to make the product right. That starts initially with the container evaluation, and that does not just start with the syringes, but also applies to the other containers. And when it comes to the syringe, for instance, an important point would be siliconization. It has a huge impact, right, on the force, and you don't want to be too high, you don't want to be too low. And let me put it like this: it makes a difference if you have a 50 milliliter syringe or a 1 milliliter syringe. Yeah. Another example would be oxygen sensitivity. So this is just an example where we had a product that was extremely oxygen sensitive. And that was, of course, also a challenge for our commercial folks, right? Because they wanted to know what is the limit during my commercial manufacturing in order to be below a certain threshold. So, and the data that you actually see here is just some graphs from the HPLC analysis, different impurities, and we saw that one specific impurity, right, is extremely sensitive to oxidation, and therefore we would use then that data then also during the commercial manufacturing to have then the nitrogen flashing at our prefilled syringe line exactly at the right level. And the last example is an interesting one when it comes to emulsion. So maybe I mentioned the word emulsion already so many times, but didn't really explain what an emulsion is. An emulsion is like a small, like nano-sized oil droplets dispersed in a water phase. Yeah? And one of the critical process parameters for an emulsion is that you need to have the right oil droplet sizes, because the oil droplets must not be too large, because they are administered IV, and that would have dramatic consequences if we actually do not get the oil droplet size right. And in that case, we know from our experience that some process parameters have a very critical influence on the oil droplet size, such as temperature, oil-water ratio, pressure, and homogenization cycles. We tested those parameters in a very systematic way and then got the sweet spot how the process parameters need to be in order to have then the right profile. And that, of course, is the same also when you do the same analysis for force of the syringe, right? Where you actually have different syringe shapes, different degrees of siliconization, where we would also use like, a DOE, design of experiment approach, in order to develop the syringes for our customers. And when you do all the development, you still need to have the scale up at the end. Because what we always saw, like, even if it works perfectly on a lab scale, it does not mean that it runs also in a 1,000-liter batch. And therefore, our pilot plant is a very nice way to do that continuous scale up with larger tanks, in order then also to have then the commercial size already before you go into the commercial manufacturing side. So with that, I would just like to summarize the main points. So I hope I could give you some idea that how we run the prefilled syringe manufacturing at our sites. So the examples that I showed you were photos mainly from Graz, but at the same time, our US manufacturing site has a very, very similar manufacturing setup. We only can at this point utilize nested prefilled syringes, with the advantage that they come pre-sterilized. We can do aseptic manufacturing, can do all different shapes that I briefly also outlined. It doesn't stop at the filling, because when it comes to the packaging, we would have then the different options of different packaging configurations. I really believe that syringes add a huge value when it comes to more complex, high-value products. So if it's a very simple, like, buffer solution, you would usually not use a syringe, because the cost of a syringe at the end of the day is actually more expensive than a glass ampule, for instance. At the same time, for a high potent API, this really adds value. We have both aseptic manufacturing and terminal sterilization implemented, and our adaptable, flexible filling line, like, with those mobile vessels, with those bags that we simply attach to the filling line, allows us then to also then meet the needs of our customers. So with that in mind, I really hope that I could give you some insight, like, what we do at Fresenius Kabi for our customers when it comes to prefilled syringes. Please also visit us at our booth. It's just like, around the corner, just a few meters and to the left, right, in that hall. And if you also would like to understand a bit more about, not just about syringes, but about IV bags, please also then join the talk of my colleague, who is going to give a talk about our proprietary free-flex bag technology that we offer to our customers to fill basically your IV truck or standard solution. With that, I would like to thank you very much for joining my session, and please visit us at our booth, right? And of course, I can happy then to take any questions then there. Thank you.